To formally get things going, I want to introduce to you the Director of the Center for Clinical and Translational Science, Dr. Becky Jackson.
Hi, good morning. You know, we need a little OHIO or something to wake us all up. As Stuart said, I'm Rebecca Jackson, the Director of the Center for Clinical and Translational Science here at The Ohio State University. And on behalf of all the faculty and staff that are involved with the center, I really want to welcome you today to one of our tools of the trade. The overarching vision of the CCTS is to really take basic science discoveries and translate those to new paradigms, new therapeutics, and new diagnostics that actually ultimately improve human health. And when we really look at this, we really think of this as a bi-directional process. So learning from the bedside to actually inform the basic science discovery, and then translating that basic science discovery back to the bedside and then into the communities for actually the adoption for changes in healthcare. And certainly one of the things that is critical for being able to do that is to really be able to access biospecimens, including DNA, RNA, tissue, and of course plasma and serum that really help to inform and allow us to do some of the basic science discovery. I've been doing this for a long, long time, longer than I'd like to admit to. And probably many of you are much like me. 15, 20 years ago, our approach for handling this really was the freezer in the back room. And so we created cohorts of patients with certain characteristics or certain diseases. We kept biospecimens across the course of those studies. We put them in our freezer, and then as scientific questions emerged, we actually went to those, withdrew them, and used those not only for our actual initial pilot studies, but in fact many times for actually our validation and as well. And the problem with that is we really didn't have the quality control. And so for those of you, for example, who work in areas like proteomics, you know the problem of freeze thawing. Or if you work with RNA, you know the problems with stability. And of course, even the problems of how you keep the tissue samples in order to be able to go back there and probe those with some of the newest techniques. And so over the last decade, really the science associated with how you manage biospecimens, how you link those biospecimens together with clinical characteristics to actually be able to pull out cohorts of individuals that are phenotyped to the level that allow you to answer your scientific questions has really emerged. And that's really what today's program is about. So what Tools of the Trade has really been developed to do is to try to bring to you new techniques, tools, and skills that will allow you to move your science forward a little bit more effectively. We try to bring forward some of the controversies, some of the gaps, including some of the gaps that actually exist in our own institution, that we can work together collectively, and to try to find out from all of you the things that you need to be able to ultimately be the most successful. So over the course of the next 4, 5, 6 hours, you're going to see a host of individuals across both The Ohio State University and Nationwide Children's who have really worked together with the Comprehensive Cancer Center as well to really try to move forward the advances in biorepositories and biospecimen management. I hope this is a fulfilling and rewarding. Feel free to really actively engage. That's what we're here for. And then at the end of this, please give us feedback, not only how useful this is, but in fact, what other types of issues that you have that we can actually move forward and address. Again, I welcome you. I hope this is a great day. And I'm actually going to turn it over to Stuart to introduce our first speaker.
Well, and our first presenter will be Dr. Nilsa Ramirez, who, as you can see, is the director of the Biopathology Center at the Research Institute at Nationwide Children's Hospital. She earned her medical degree at the University of Puerto Rico School of Medicine, and she also serves as the director of surgical pathology in the Department of Pathology and Laboratory Medicine and an associate professor of clinical pathology here at The Ohio State University. So, Nilsa.
Okay. I thought that it was a good idea to look at this topic because when you look for specimens, you look for resources, and many of you, I am, confused. Where do I go? Do I go to a tissue bank? Do I go to Biorepository? Do I go to a specimen resource? And there are too many terms that are applied to these biological collections of basically human, animal, plant, or microbial samples. And I just listed some here. As you can see, the list is longer than this. But these are things that I'm sure all of you have seen as you look into resources to use. You can have those that address the issue of biospecimens, or specifically using the word tissue, or those that are more specific to a disease, for example, a leukemia bank, GI tissue repository, brain specimen repository. And a term I came across recently, biological resource center, that is very popularly used in Europe. So what do they all have in common? Where they bank or store specimens. And again, you use, what do they store? Biospecimens, specimens, tissues, tumors. So as I navigate the world of biorepositories and biobankings and tissue banks, it has been very clear that there is a difference. When I think of a tissue bank, I think of a simple operation. This is when I think of somebody's freezer in their lab in the back of the room. Yeah, go and get it there. I have some specimens that you can use. Versus a biorepository, which it is technically a more complex operation. When you have the biorepository, you not only have procurement, processing, banking, sample quality assurance, and quality control at a very high level. Also, they offer analysis, distribution, and data, which is very, very important. You can have many services, including various storage modalities, informatics, and this informatics is just not going just to support the collection of data, but the inventory, the interactions with whoever is sponsoring the collection, and with the investigators. You also have services for virtual microscopy, histology, molecular genetics, and customized kit management. Size, we think of a large operation. This is a very relative term because you come to me and you say, I have a large amount of tissue that I want you to bank, and I always ask, how large is large? Are you talking about 2 million specimens, or are you talking about 150 specimens? And 150 specimens may be a large collection for somebody, but it's all relative. So a tissue bank, those of us that deal with this on a more regular basis think of a more simple operation where you do specimen procurement, banking, and distribution. And that's basically it, just the storage of the specimens. If there was a recent article in which a lot of people that are very well-versed in biorepositories were asked about the best term to use, and biorepository and biobank appear to be synonymous to those that are in the field. Some people think that a biobank is something in which you store the specimens and you get them out in a relatively fast, like just storage, temporary storage, versus a biorepository being something in which you store the specimens and they may be there for a prolonged period of time, either because of the way that they were collected or that the data associated with them needs to mature, or things like that. But about 62% of those that were asked about terminology thought that biorepository and biobank was basically the same thing. So what type of support you're going to get? Usually they're going to have different storage modalities, and I'm going to go over that. Inventory, they need to know what is there, and some degree of quality assurance, and again, large or small size, but this is sometimes when you get into the more of a relatively small amount of specimens being collected. The organizational structure, well, they can be, this types of units can be part of a large public or private group. For example, they can be based in a university or a hospital, a pharmaceutical company, or they can be freestanding. And here, so you can get an idea of the structure of a biorepository, you will have a director somebody with a program manager. Usually this is a very experienced person that knows the area very well. Could be an investigator. And then over there you will have all types of managers that manage the different areas of the biorepository, and also the laboratory technicians or the investigator services coordinator, which will be the person that interacts with the investigators that are looking for the materials that are stored in the biorepository. Regarding personnel, you have an incredible variety of people with different backgrounds, MDs, PhDs, people with advanced degrees, histotechs, medtechs, computer scientists, college students, sponsor project officers, accountants, and you can have over 100 employees. Actually, at the Biopathology Center, we have 140 employees. So it could be a large operation at that sense, or you can have a smaller operation, which will just be along the lines of the tissue bank, in which you have a director. Sometimes you have a manager and a laboratory technician. Sometimes you have the director, and the manager is also the laboratory technician. So you have 2 people at least that deal with this, and they can have different training. And usually you see anywhere between 3 to 10 employees in that setting. Governance. The governance of a biorepository is incredibly important, because these are the people that are going to not only decide on the structure and how things are going to run, they're ultimately the ones that have custody of the specimens or the biospecimens. They're the ones that are going to decide who gets the specimens. And this is consistently done based on the scientific value of the proposals that they receive. And that can be structured depending on the intent of the biorepository as it was originally designed. So for the level of the biorepository, you're going to obviously have roles that will deal with receiving, banking, inventory, and distributing the biospecimens. There should be a method of establishing quality control and quality assurance standards and procedures. Promotion of the biorepository resources as services. You can have a wonderful biorepository, but if people are not aware of it and they're not using it, to quote Laura Monovich, it's not a biorepository, it's a museum. So you really have to, I love that statement. You really have to be aware that things need to move. We really want these things to be used. Nobody is just trying not to give you the specimens. We want you to use them, but they have to be used wisely, because they're very precious resources. Think about the fact that almost all those specimens are donations, that patients donated that, so we can advance science. So you have to be very conscious of everything that comes, that has to happen for that biorepository to exist. And they will also be, in the biorepository, supportive of establishing an informatics resource. In this model, I have introduced the utilization committee. This is a layer that sometimes, if you don't have a large resource, you may not need. But sometimes it's very important, especially if you have a lot of clinical data associated with the biospecimens that you're collecting. They will be the ones that provide the oversight of the biorepository. And who's going to be there? Well, you will need scientists. They could be basic scientists, clinicians, and biorepository staff. Hopefully the director and the program manager, or somebody at a high level of the biorepository that can interact. They are the ones that are going to develop the policies related to how the biorepository is going to work, prioritize and support the banking and distribution, and be sure that the right amount of specimens are ready to be used by the investigators. Then you go into the oversight committee. They're the ones that are going to be over the whole operation, including the biorepository and the utilization committee. They are the ones that are going to direct what type of science and translational research efforts are going to happen. They're going to develop any protocols, any clinical protocols. They're going to be aware of what's going on and what is it that the investigators need. And be sure that the results are published. That's another issue that we have. We deal with releasing the specimens. Everybody's happy. The investigators got the specimens. 5 years later, where's the paper? Oh, well, I don't know. We really need to validate the resource. You need to show something. And the way to show it that we have right now is by sharing the wealth, sharing what you encounter when you use those specimens, and publications is at this point one of the most important tools that we have to assess the value of this. So this is not just, oh, I got the specimens, I did something, and I published the results and I shared the results. And they're, again, the ones that decide, based on the scientific value of a proposal, if an investigator is going to receive the specimens. This is more of an institutional group. In a setting like Ohio State, it will be somebody more at the level of, if you're dealing with a department, it will be somebody at the level of the department, group leadership, and then a multidisciplinary advisory board members. If you have a proposal that is outside of the level, the type of science that your group does, you can always have ad hoc members from people from other fields that will come into the area and advise on specific projects. So just the fact that you don't know a lot about that, or you don't feel comfortable about whatever they're proposing, you can go into the system and see who over there can come and assist you in that particular thing. As a pathologist, I get calls sometimes in settings like this. Hey, Nilsa, there's a lot of pathology involved in here. What about, the time frame that they're putting to do this immunohistochemical stain? Sometimes it just has to be something about the format that they're putting things. Is this doable? How much this is going to cost? So a lot of times you do need help from other resources, and it's a good idea to be open-minded about having that. So again, you are going to have overlapping of membership in the three entities because there has to be some continuity. People need to know what is going on at each level. There are a lot of things that need to happen to put together a biorepository. And it is not uncommon for people to come and call me or ask me, I want to start something. What do you think? This is what I have. What things do I have to keep in mind so I can come up with the resources to put together this biorepository for my department? Or I am not happy or satisfied with the specimens that my institution is collecting, and I want to start a collection. What do I need? Well, first thing you're going to need are resources, cost. It is expensive. So sometimes people come and say, I want to do this, and then my first question, and those of you that deal with me, is who's going to pay for this? And you need to have a budget. If the institution or a sponsor is going to start this, if you have a group or some foundation that wants to give you some money to start something, you need an annual budget. And it all depends on the size and complexity of the operation. Again, operations may be small, but if you are going to do stored DNA and RNA and frozen tissue and slides that are vacuum sealed, that costs a lot of money versus having a larger operation that is just going to store serum. So you have to be aware of what is it. You have to have a plan for supporting the growth and the new technological developments, not only in the fields of science that you're dealing with, but in the storage mechanisms that are available to biorepositories. Are you going to have the money to buy the latest, greatest freezer that came out that is really the one that is going to fit your needs, versus using something that consumes a lot of electricity that nobody in your system wants to take care of every time it breaks? So you have to think about those things. If you're an investigator, are the services that are going to be provided to me as an investigator going to be free of charge? Or is it going to be a fee for service for some of the services that are going to be provided? For example, if you need an amount of money to start a bank, you need $50,000 to start a project, and you know that every year you're going to need $10,000, and all you have is 5 at your institution, where are you going to get the other 5 from? Well, I'm going to distribute the investigator the specimens, and I'm going to charge a fee. You cannot sell specimens. Please don't say that you sold the specimens, because that's illegal. You are charging a fee for the service of taking the specimen out of the unit and packing it and giving it to the investigator. And that's what you have to come up with. And when you look at the courts, for example, here at Ohio State and Nationwide Children's, they have fees for charge. And that's exactly what the biorepositories do. They charge you a fee. They generate program income, and that assists them in putting together their services. So again, sometimes if you don't have enough resources, think of that type of arrangement. Management, you need somebody to be in charge of it. Many times, let's have a biorepository, let's call pathology and have somebody from pathology take care of it. And the poor person in pathology already does service 100% of the time, and he or she are in charge of the biorepository, and they don't have time. So you have to have a dedicated professional that is going to see it. Doesn't necessarily have to be an MD or a PhD, but somebody that is going to be dedicated to anything that happens that needs assistance of. You need to establish professional standards. These are the criteria I want on somebody that is going to be working at the biorepository. You have to develop standard operating procedures in writing. You go, I'm the chair of the Biorepository Accreditation Program for College of American Pathologists, and you're going to, sometimes I go to these inspections, and do you do this? Oh yeah, we do it all the time. Where is it? Well, come here. You were here 3 years ago when we started. What is it that we do? It's not in writing. If it's not in writing, it doesn't exist. So you have to write things down. It doesn't have to be pretty, but you have to write it down, and there are formats for that. Best practices for biorepositories. Here in the United States, we're very familiar with the ISPR and NCI best practices, but in Europe, there are a lot of best practices developed by very nice groups, very similar, all of them, that are available, 14 or 15 different sets of best practices for biorepositories that are available internationally that you can always look. You don't have to do everything. There are best practices. They're just telling you this is what you need to do. And it is of incredible help. It allows you to organize your thoughts and put everything in perspective. I'm going to be able to monitor my freezers. Am I going to be able to monitor my freezers with a backup system that also has a third backup system? That costs money. And all these things are available when you look at these best practices for biorepositories. Location, where is this going to be? Is it going to be in a single institution? Are you going to have it in campus? Are you going to have it spread all over the institution? Or are you going to be part of a network? Is the network local, regional, or in state? Is it national or international? Are you going to have a combination of your institution plus a network? Are you going to keep your own bank? Let's say Ohio State is part of a network, but you have to have your own bank. Then you have your bank, but you also have the services that you integrate with the network. So all these things need to be, again, clear before you start the putting together the biorepository. Is it going to be a central collection? Are you going to collect from all of these places and they're all going to come to a certain point, or everybody's going to keep the collection where they're doing it and you're just going to have a virtual biorepository in which a central location knows what's going on, knows what the inventory is, but the specimens never come there. They're kept somewhere else. So that is another setting that you can entertain, especially if you don't have a lot of resources. Transportation, how are the specimens going to move? Are you going to use a courier? Is it going to be an institutional courier, somebody from a courier system at Ohio State versus using FedEx or using any other commercial group? Remember, there are state and interstate regulations for moving biological products. So you have to be aware of that. There are international rules and fees. Oh, I went to a meeting and I met this guy I trained with, and he's now practicing in India, and he said he's going to send me 50 cases of this. Really? How are those going to come through customs? Do you know that there are certain countries that have a ban on exporting biological samples? So before you do that, clear out. It's nice to have to see all your friends you trained with when you were a resident, but be sure that that arrangement that you're so excited about can really happen. Transportation, there are regulations, for example, the International Air Transportation Association, or IATA, shipping regulations. And if you are going to be dealing with moving specimens in and out of your system, your employees that pack these things need to be aware of this. Banking, different modalities. Are they going to be frozen? Are they going to be in room temperature? Do you need to have control access to the biospecimens? So they have to be in a secure place. Now, to be secure, you probably need an institution that have, your key cards, that everybody knows who goes in, temperature monitoring system, motion sensors. If they're going to be in a secure structure, are you going to have rooms or a floor or a building? You have to clarify all that before you start collecting. All these things have to be said before you start. Processing. How are you going to process the specimens? Because you're more complex than just the freezer in the back of somebody's lab. So are you going to be affiliated with cores and other institutional or outside clinical research laboratories? What type of testing you're going to do? And one of the most important things, the quality of the specimens. Are you going to have a system? And how is that system going to work? Are you going to evaluate the specimens when they come in, when they go out, or only when they go out? Do you know what you have in your freezers? Is it all good quality? All of that needs to be assessed. And you need a network of people because you need certain professionals to evaluate the quality of certain specimens. For example, tissue, if you're going to do tumors and you need your tumors to be 80% tumor, less than 20% necrosis, you call the pathologist. You call somebody that can look at it or somebody that is trained to look at that particular area. So all of that, again, these are things that come into having a functional biorepository. Then be sure that the distribution of biospecimens to investigators follow the requirements of the project. Every project has a different set of requirements. Somebody will come and say your pristine specimen, more than 80% tumor, less than 10% necrosis. Somebody else will come and say, I'll take whatever you have. Somebody else will say 50% necrosis is fine, 10% tumor is fine. So whatever fits for one investigator doesn't fit for the other. And you need to be sure that you're following those requirements. Informatics, incredibly important and incredibly expensive. Data collection and distribution. Inventory, need to know what you have, where it went, where is it? Because whoever is paying for that biorepository wants to know where the stuff is and at what stage. Is it frozen? Did anything happen to the specimen? Virtual microscopy is a fantastic tool when you want to keep costs down on not having to use FedEx or not having to use transportation or worry about it. Then you scan the slides and make them available to those that need to review them. And I'll go quickly over how we do it at our Biopathology Center. And the interactions that you're going to have with the sponsor or the governance of those that are affiliated with you. For example, for the cancer cooperative groups, they have statistical and data centers. And they are the ones that send a list to our bank and say, these are the specimens that you're going to distribute. Then who's going to interact with them? Are you going to provide them on a daily basis with the data that they need? Are you going to tell them how the inventory is moving? Regulatory issues. There is a College of American Pathologists laboratory accreditation program, and there's also a biorepository accreditation program. Are you planning on also testing for patient care? Then you have to be looking at the laboratory accreditation program. Are you just going to be a biorepository? Then the biorepository accreditation program. Do you need CLIA certification because of what you want to do with your specimens? Who's going to deal with the material transfer agreement or material use agreements? There are, of course, HIPAA regulations that we have to follow. Who's going to keep track of the informed consents? who's going to keep track of being sure that the informed consents are updated. If you're dealing with children, are you going to reconsent after they turn 18? So all of these things need to be taken into consideration when you're putting together a biorepository. IRB issues, are you going to be dealing with a single IRB, or are you going to be dealing with multiple IRBs or multiple institutions? Disaster recovery initiatives. Sadly, these things happen. Are you going to have a remote facility to store part of a sample, which means that if you had 10,000 samples, now you have 20,000 samples, and 10,000 of those are going to be at least 30 miles away from where you are. So who's going to take care of that? You have to duplicate your services. What happens if you lose the biospecimens? I think about that sometimes. Keeps me awake at night. The data, is the data going to be duplicated? Where are you going to store it? How much is it going to cost you? Where is the data? Is it with you? Is it with somebody else? Security measures to prevent loss. Any type of insurance that you need in your building, in your structure, who's going to cover the insurance? Your institution or the sponsor of the project? Flood insurance, whatever type of insurance it is. You have to be, things to remember. Personnel, how much is going to cost you? Are you going to have full-time employees or part-time employees taking care? And if you're going to have part-time employees, be sure that the part of the time that they're going to invest in the biorepository really happens. Because I know how it works sometimes. Oh yeah, I have 3 part-time employees. Those employees don't even know where the building is. So it's very important that people are aware of the regulations, aware of what you're doing, aware of the biorepository. Training, education. Are they going to be educated? Are they going to have access to meetings like this, in which they can go and see and talk to other people that do what they do? Are they going to participate in any type of activity that the sponsor has? And are they going to travel? It is amazing that you don't consider that, but if you are part of a network, there is some traveling involved. Are you going to travel once a year? Who's going to travel? Who's going to pay for the travel? And then, very, very important, we get all excited. We have this collection. What's going to happen to it when the funding stops? You have 3 million specimens. They're clinically annotated, mature data. They're wonderful. The NCI cut your grant. Where are all those freezers going? Who has custody of them? You solve that ahead of time. Before you put your biorepository together, you have a plan. This is what's going to happen. Every once in a while, I get a call. So-and-so retired. Nobody wants to take care of their collection. Do you want it? When Dr. Scott Jewell was here, I worked with Scott for many years. Constant thing that Scott had to deal with. People call, these things happen. So think about what's going to happen. It's like your children. What's going to happen to them when you're not around? You think about this the same way. You have to be sure that these things are taken care of, because there are people that are going to benefit from this. So I'm going to use as an example of that functionality that you can have in a biorepository, for the time I have left, some of the things I'm involved with, especially with the Biopathology Center at Nationwide Children's. Our Biopathology Center is largely federally funded. And originally, and it was always been our objective to procure, bank, process, and distribute a large number of biospecimens and related clinical and pathological data, very important, for the purpose of supporting cutting-edge research. This was started in the late 80s by the late Dr. Steve Qualman, who was the chair of pathology at that time, Columbus Children's, and also renowned rhabdomyosarcoma investigator. And the main effort was to support pediatric cancer clinical trials. It started by doing banking for the predecessors of the Children's Oncology Group, and also with the pediatric division of the Cooperative Human Tissue Network, and later doing this today's group of presentations, this topic is going to come up about what is the CHTN and how you can get specimens from that. We have many projects that are supported by our Biopathology Center. Some of our core grants, or our largest grants, are the cancer cooperative groups, energy oncology, which is recently a group that recently resulted from the merger of several groups, including the gynecologic oncology group and the children's oncology group and SWOG. And those of you that are in the clinical areas here at OSU are likely familiar with both SWOG and GOG. The CHTN, and a lot of other efforts, many of them under Dr. Julie Gastar-Foster, like the Cancer Genome Atlas Biospecimen Core Resource, the Target Initiative, the Berkeley Lymphoma Genome Sequencing Project, and the newest Exceptional Responders and the Alchemist study. And we recently started being the banking facility for the TCC initiative from the James. We also have in-house projects, and this is just a list so you can see that we have different. We do have banks from cardiology, ENT, and pediatric brains, and then we also have other groups. This one here, where is the Ohio Perinatal Research Network is one of our projects, and several investigators receive specimens on a regular basis from these initiatives. Specimen collection. What we have is a centralized mode, meaning specimens come from the outside. So we receive specimens from Nationwide Children's and from more than 500 national and international institutions. And these specimens include tissue, and the tissue comes as fresh, frozen, fixed in formalin, embedded in paraffin, blood and blood products, bone marrow and urine. And we QA the biospecimens, including the paperwork upon arrival. It is very important to be sure that what is in that form that comes with the specimen is actually what you receive. And if it's not there, you have to immediately contact the institution and be sure that they know that instead of getting 3 tubes of blood, you got 1, or that the 3 tubes of blood that you receive are broken. All of that needs to happen immediately. That's very important. It doesn't sound like some earth-shaking type of scientific research, but it is very important that you can allow for that. And our courier is FedEx. We get shipments from Monday through Saturday, and we get between 70 and 100 packages a day, which comes to about over 1,000 specimens that we receive in our center a day. We do clinical trial support. And when you do clinical trial support, you go to the sponsors or those when the clinical trials are being developed, and you interact with the group that is being it. So you get input. Your biorepository staff and the investigators from the group need to talk to each other. It is very sad when we get a protocol that has already been approved by everybody and needs to approve it. And when we look at it, it's like, we cannot do this for them. Who came up with this? Nobody, we were not asked. It's either a really expensive proposition or something that doesn't fit the type of mechanism that we have already established for a sponsor. So if you are going to be working with somebody else, you need to sit down with that investigator and be sure that he or she understands how you work, so the specimens can be collected properly. You have to be able to provide cost estimates for projects. Okay, I'm going to charge you a fee for service. How many specimens are we talking about? When do you want them? That is all very important. We also provide information to them about collection supplies and containers. We, for example, develop and produce these laminated posters or placemat-like posters that they can take to the operating room and see how is it that the specimen is going to be collected, and also collection videos. So again, there are right now with the internet, and there's so many things you can do to communicate with those that are far away from where you are and are collecting specimens for your biorepository, and to how do you want things to be done. Kids. We provide this kids for over 30 protocols for these groups listed here. And each customized kit will have materials that are needed specifically for that. We have a kit ordering process that was developed at our institution and by our group at the Biopathology Center, in which the CRAs can go in and order the kits, and then they're ordered, they get processed, and they get sent to them. So when the patients come, they have the proper kit to provide to the physicians so they can collect the specimens and send them back to us. You can have dual chamber kits if you are going to allow for the shipment of frozen and room temperature biospecimens in one shipment, or if you're just going to do all biospecimens shipped at one temperature. Just putting stuff in a box doesn't work. If it's winter, and they were not supposed to be frozen, they may arrive frozen if they're not properly insulated. If it's summer and they were not properly insulated, then rather than having frozen tissue, you just have a mess of whatever they embedded it in all over the box. So you have to be also aware of where in the country you're sending the specimens and what time of the year you are doing this. So it is important to have some type of method in which you can provide them at least either with the material or with instructions, proper instructions as to how to pack the specimens. And that will apply for something coming from Australia, where we do get specimens from Australia, or for something coming from here to Nationwide Children's, or from Nationwide Children's here. You have to be aware that anything that's going to be outside of something being hand carried, you have to be able to let them know how to transport the specimens. Storage modalities, for example, in our setting, we have over 4 million biospecimens stored. We have frozen biospecimens that could be in -80 freezers or liquid nitrogen freezers. We have a re-scientific monitor system for monitoring the temperatures, and we have also backup freezers. We can store the formalin-fixed paraffin-embedded tissue at room temperature or at 4 degrees. And we have ways of also storing any glass slide cytology preparation. For processing, we offer basically, these are basic things that you can find in any core service. For example, processing and recording of blood and plasma, embedding tissue, LCM, macro dissection, creation of tissue microarrays, assays in the cores and the for genomic extractions. We have a workflow relationship in our setting with our cores. And this is something, again, that you can establish. You don't necessarily have to reinvent the wheel. If your institution or your system or your department has resources, you can always integrate the biorepository with the resources that your institution has, or those resources that are available to you or to the network that you're participating in. Regulatory things. You have to be aware of having the capacity to track any documents associated with a project. We are accredited by the CAP Biorepository accreditation program, so that allows us to have our SOPs arranged in a certain format, and it really is a very nice way of looking as to how your biorepository operates. We also have COG reference laboratories under Julie for ALL, Wilms tumor neuroblastoma, and those are accredited by the lab program and certified by CLIA for patient-related testing. And our morphology core is also CAP lab accredited. And this is important if you're going to do samples that are going to ultimately result in any type of testing that is going to be involved in patient care. So those things you have to look into when you're putting together your setting. We're involved with the regulatory efforts of anyone that is banking with us. And many of the leadership in our Biopathology Center are members of ISBER, and all of our employees are trained CITI. Informatics, we use a system that was developed at our Biopathology Center. It goes by the acronym of STARS. And this is an enterprise system that facilitates best practices related to biospecimen management and distribution. And basically what you have is the life cycle of a specimen. We know when it was accessioned all the way to the point in which the specimen was distributed, if there is any reporting, and if there's any auditing that needs to happen. Also have the capacity to provide for institutional performance monitoring, any interactions with any of the sponsored document imaging and managing system, and the kit request and tracking program that I mentioned. Digital pathology, many of you are familiar with this. This is the technique of digitizing tissue glass slides. And these images can be then exported and reviewed, managed, and analyzed electronically. Has a lot of advantages, especially reducing the time that pathologists take for review. If you have to put in, you're sending something to California, then it has to come back, versus somebody gets on the system, reviews them, and gives you the results, and immediately tells you what you need to know. It also reduces the cost associated with distributing the slides, and allows you to have a digital archive of the material, and also allows you to develop some image processing and analytical tools. Regarding equipment at this time, we have 8 whole slide scanners, 1 oil scanner, and 1 fluorescent scanner with the capacity to scan 840x images in 24 hours, or 2,520x images in 24 hours. Again, it all depends on how many images you're going to do and how fast you're going to need them. We have our virtual imaging and pathology and education and research. This is a system that will provide pathologists with necessary information to do a digital review. And this system you will go in, it will advise or notify the reviewers where the cases are assigned, and also notifies the Biopathology Center where the work has been completed. And we have many users, mostly pathologists, right now in 16 countries outside of the US. Freezers, we have 60 liquid nitrogen, 62 minus 80 mechanical freezers, and these collections grow at a high rate. So that is, again, how you need to be proactive and be sure that you know how many freezers you're going to need in what particular time. We have an off-campus facility that will eventually be able to house 133 freezers with the capacity to hold more than 5 million biospecimens. So this is all done with plans in mind, and the idea that future growth needs to be addressed. We recently have a 6,000 gallon vacuum jacketed liquid nitrogen bulk tank, and that allows us to get the liquid nitrogen directly to the freezers without having to go into getting the liquid nitrogen delivered on a regular basis and all these things. So all of this is just an example of planning for the future, and being sure that you can accommodate whatever you expect to happen. This is just, again, from our Kinnear facility, where we maintain 46 minus 80s and 17 liquid nitrogens. And this is from our hospital, one of our hospital facilities, in which we have 8 minus 80s and 41 liquid nitrogen freezers. Distribution, we distribute specimens following a proof of scientific proposals. We do QA before the specimens are distributed. We're in constant communication with the investigator, and we are sure that our employees know how to properly send the specimens out. We do use FedEx and get some discount from all the stuff that we do. So this is how the work flows. Specimens come from the institution. They go to our central processing. If they need molecular genetics or any type of testing, the results are then go back to the institution if that's the setting that was agreed upon. Or they can go to the course, for example, the morphology course that will create the slide for review. The diagnosis is confirmed, and the specimen goes back and gets processed and banked. Or if the specimen is distributed, We always are sure that, of course, the distribution is approved by the sponsor and that there are all agreements need to be, and there's IRB approval documentation and all these other things that need to take care. And I think my last thing, publications. We encourage everyone that is going to be using the biospecimen to publish at a scientific level, but we also publish about the things that we do and about the things that are important for in the biorepository world, what we need, what we learn from what we're doing. For example, anything that has to do with administration of a biorepository. In a recent book, Developing and Organizing a Biospecimen Repository, I co-authored this chapter with Laura about management and key personnel of the biospecimen repository. And also in a recent publication, this one came from the CHTN, and there are many people from Ohio State, like Dr. Ayers, Dr. Lori Johnson, who I just saw sitting back there, Laura Monovich, and Jay Bowen from Nationwide, myself. And we participated in this. This study took over 10 years to be finalized, but it was just a very wonderful study on the effects of frozen tissue storage conditions and the integrity of RNA and protein. And also Dr. Qualman, Dr. Scott Jewell, and the late David Newsom contributed to this. So again, just be sure that when you're putting together your biorepository or when you're participating in any type of biorepository-related effort, that all these things or most of the things are taken into consideration. Because we all have great ideas. I really enjoy getting some new investigator or new faculty member at Nationwide coming with ideas about a biorepository. But as soon as I say, who's going to pay for this? Do you have an idea how you're going to distribute this? What's going to happen when the funding ends? You just see the change in their face, like, oh my God, I didn't think about all that. And they're so depressed when they leave. But I think that we all have these wonderful resources here that we can use, and I hope that you use them. Thank you.
Nilsa might have time to answer one or two questions while our next speaker gets set up.
Yes.
I don't know if you can you hear me?
Yes, I can hear you.
So you provide a lot of really nice services, and for someone who might be interested in using some of those services, how would you like us to contact you, or what is the best way to get in touch with you to figure out how to go forward?
It all depends on what type of services. For example, I get a lot of inquiries about starting a collection. Like, I'm going to do something in my department. Can you bank for me? And that we do it through our core. So there is a website that we can provide, you know, and all of this information is going to come a little later regarding how to contact our core. And many of the services that this audience will need will come through our core, biopathology core. And you can contact us that way. If it's something that somebody wants to just run it by me just to see, you know, I have this idea, what do you think? People usually send me an email, and we make an appointment and meet for, you know, half an hour. I take people on tours, or you can tour our facilities too. Sometimes people just want to see what we have. So,
Thank you.
okay, thank you. I'm going to be around, so if anybody has any questions.